All right, so now I'm going to be taking you through the process of mitosis. So mitosis, the purpose of mitosis is to create two identical nuclei. And remember that in the nucleus, that is where the DNA is. This happens after G2 of the cell cycle, so after the cell has copied its DNA and it has prepared for division. And it occurs in four stages. There's prophase, metaphase, anaphase, and telophase. So we'll take a little bit of a closer look at each of those stages. So just remember that all cells spend most of their time in interphase, right? So G1 was when the cell was growing and it was maturing in size, performing all of its regular activity. If it gets a signal to divide, it will enter into S phase, where all of the cell's DNA is synthesized, where you're going to make basically a copy of all that cell's DNA. And then you enter into G2, where the cell doubles in size, makes copies of its organelles, and makes those centrosomes so that it can divide once it is ready to do so. After interphase, then we start the mitotic phase. Okay, so this is a picture of interphase. Our DNA is doubled, but it's still in that loose chromatin form. The nucleus is still there, but those are our special structures that we're going to need for division to happen. But everything else is ready to go into mitosis. So we start mitosis. The first stage of mitosis is prophase. So interphase is not a stage of mitosis. Prophase is the first stage. During this time, the nucleus is going to start to break down. So we're going to start to break away the nucleus because we have to be able to move the chromosomes around. Spindle fibers are going to begin to form. These are special fibers that are, their job is to move chromosomes around. So we'll have those form. And then our duplicated chromosomes are going to begin to condense. So right before this, remember that they were in chromatin form, and now they're going to condense into that duplicated form where they have the two sister chromatids. Okay, so this is showing you a picture, right? Remember, we started at interphase where we still had chromatin. We had doubled all the DNA, but it hadn't condensed into chromosomes yet. So in prophase, we've now condensed into chromosomes. Nucleus is going away, and we have some spindle fibers. From prophase, we move into something called metaphase. In metaphase, the spindle fibers have moved the duplicated chromosomes right along the middle of the cell. So it's going to line them up right across the middle of the cell. This is important because we want to be able to separate those sister chromatids that make up the duplicated chromosomes. We want to be able to separate them evenly. And so by lining them up, that makes it a lot easier to do that. I always remember that metaphase is middle because they both start with M. Okay. Next, so here we have, again, our interphase into prophase. So from prophase, we now see them lined up across the center of the cell. And then we move into anaphase. In anaphase, these spindle fibers have attached to the chromosomes, right? And they're going to actually pull the sister chromatids apart, and they're going to drag them to opposite sides of the cell. So they work at just pulling those apart. So now they are no longer duplicated chromosomes. They are now single chromosomes because we've separated those chromatids from each other. And a lot of times I remember anaphase and apart, right? So the chromatids are being pulled apart, and they both start with A. After anaphase, we move into our next stage, which is telophase. Telophase, a couple of pretty important things are going to start to happen here. So first, we're going to start pinching in the cytoplasm of the cell. This is something called a cleavage furrow. A new nucleus is going to start to form around both sets of DNA that were just separated from each other. The spindle fibers are gone. You can no longer see them. And the chromosomes are starting to unravel, and they're going to end up unraveling back into that chromatin form. At the same time that telophase is happening, cytokinesis is occurring. In cytokinesis, you're going to actually split those two cells completely. So you end up with your two identical daughter cells at the end. And the chromosomes have completely unraveled back into their chromatin form, and they pretty much look like they did back in interphase. And that's that complete cycle. So you went from one cell that looked like this to now you have two cells that have that same appearance, same DNA, same chromosomes. So they are identical to each other. All right, so this just shows you the entire cycle. Again, we went from having chromatin, we condensed to chromosomes, they lined up, we separated those chromatids from each other during anaphase, and then in telophase, they unraveled, new nucleus, and then we divide those cells in half completely. All right, this is what it might look like in a real cell. So this is a plant cell, right? So they're showing you interphase here. This is prophase, so it doesn't look quite nice and neat the way that you'd think. This is metaphase where they're lined up. Here you can see those chromatids pulling apart from each other, and there your telophase where your new nuclei are forming. So sometimes it's kind of neat to see them in a real cell. All right, so why is mitosis important? Why are we learning about this, right? So we need mitosis in our body for the formation of our somatic cells. We need new somatic cells, our body cells, for growth and for repair, right? If you get a cut, you need to be able to repair that cut. You do so by making more cells. You grow. You are not the same size that you were when you were born, right? You grow by adding more cells. That's mitosis. There are also some unicellular organisms that will use mitosis for asexual reproduction. Organisms like protists and fungi, they can do this. So they don't reproduce sexually, they don't make gametes. So instead, they just make copies of themselves by doing mitosis. They have to do mitosis because they're eukaryotes, which means they have a nucleus. All right, so does mitosis happen in prokaryotes? Okay, so prokaryotes don't have a nucleus, so they can't do mitosis. They instead do a process called binary fission. And in this process, they just make a copy of their one chromosome. So prokaryotes have only one chromosome. They just make a copy of it, and then they split the cell in half. This is a lot faster of a process in bacteria than it is in any other cell. So our fastest reproducing cells in our body are our skin cells, which take about 22 hours. There are bacteria that can go through this process every 20 minutes. So it's a pretty big difference. All right, and then finally, we're just going to do a quick comparison of mitosis and meiosis, really thinking about the end results of this. So remember, in meiosis, you're going to get 4 genetically unique haploid cells that are called gametes. And mitosis, you're going to get 2 genetically identical diploid cells, like somatic cells. So here in our meiosis, we've got 2 cells. On one cell that's split into 2, and then we split them again. So we started with 4 chromosomes, and then we end with 2 chromosomes in each. So they are now haploid. Whereas in mitosis, again, we started with the 4 chromosomes, but at the end, we still have 4 chromosomes, and they are all identical to each other.